It's the review of the whole schmear. Okay, so the plan is just review the whole class. Now, I may not make sure this is on. Yeah. So I may not ask you questions on everything that we went over in the class, but we'll sort of hit the main points. I haven't made the final up yet, but we'll sort of hit the main points. So, and then I marked the tests so that and I posted the scores and everything, so you can take a look at that. Okay, so the plan is the whole course in 1 hour. So the first thing we'll do is the membrane potential. So we'll have 4 columns. I won't erase anything. We just have 4 columns. So we started off with the membrane potential, or sometimes it's called the Nernst potential if you're just doing it for one ion, and then it's also called the reversal potential, and that was because if you're above this potential and you open the channel, the ions will flow to get you back to the potential. If you're below the potential, they'll flow the opposite direction to get back to the potential. That's why it's reversal. And that was all related to the resting potential, which is, as we saw, mostly due to potassium. So I almost always have some question about this, and you can memorize the Nernst equation, but it's easier just to figure out basically start at 0, always start at 0, and then you have a channel that's a selective channel, only lets one kind of ion through, and the direction it will go is just dependent on the concentration. The trick that people often get confused is it's going to be the opposite. It's typically going to be the opposite of the sign of where all the ions are. So the potassium is a positive charged ion. There's more of it inside the cell. So if we start at 0, more of it inside the cell, but still start at 0. So there's more pluses potassium, but there's going to be other pluses outside to make it all 0. Then open the potassium channel. What happens? A little bit of potassium leaks out. It's positive. That makes the inside negative. So there's more potassium inside. That goes along with a negative membrane potential. So you can do the same thing for more negative outside, more positive outside, more. There's 4 different combinations of positive and negative. So that's the Nernst potential. And so I never make you calculate the actual natural log. It's more just to get sort of a feeling for like what's going on here. It just has to do with diffusion being balanced by the buildup of charge. So the membrane potential channels are open all the time, continuously open. Some potassium goes out, but eventually the inside gets negative, and that makes the potassium get attracted back in. And so then there's a balance point, and that's what the Nernst potential tells you. So that was the membrane resting potential. Then we went to the action potential. And what went with the action potential were voltage-gated channels. Voltage-gated channels. And those are ones that open up so the membrane potential channels, the resting potential channels are always open, so they just stay open. These are ones that are actually gated, so they can open or close, and voltage is what opens them. And so we had the voltage-gated sodium channel, and that was the one that gets inactivated, which means it gets open when you depolarize. So if you raise the membrane potential, depolarize, it opens it. But then even if you keep the membrane potential depolarized, it closes anyway. That's what the inactivation business is. So it's kind of a self-closing of the channel, even though the voltage is appropriate for opening. And that's the thing that really makes the inactivation is what makes the spike go in one direction, because as the spike is going along, there's a trail of inactivated channels behind it, and those guys can't open, even though they're depolarized. So that's the trick there of how the spike goes in one direction. And then there was also the potassium channels that sort of make the inside more negative, but that's not the main reason that the spike goes in one direction. It's mainly the inactivation of the sodium channels. And then the other thing that we talked about here with these voltage-gated channels is that they're also on dendrites. So normally you think of these things as being on the axon to cause the spike to move along, but they're also on dendrites. And why are they on dendrites? They're on dendrites because the cell has to kind of adjust itself all the time. So, like maybe you're taking some particular pharmaceutical drug and your brain is going to immediately try to cancel out the effect of it. If it's a pharmaceutical drug that turns up the GABA channels, well, then your brain is going to say not enough excitation, because the GABA channels are inhibiting things, and so I'm going to add some excitation, or I'm going to turn the GABA channels down. So the whole system is constantly tweaking itself to make sure that you're not epileptic or dead. Those are the two, no spikes, you're dead. Too much activity, then you get seizures. And so you always want to balance things in a narrow range. And so there's tons of these voltage-gated channels on the dendrites, and largely what they're doing is they're just sort of shaping things, keeping everything sort of like in a sweet spot, which is life. So that was the action potential. Then we talked about post-synaptic potentials. Synaptic potentials, and those guys were neurotransmitter gated. Neurotrans gated. So those guys open up when you have a neurotransmitter release. Neurotransmitter release sticks in a little pocket. That's what opens the channel. And so these guys, we talked about four main ones. We thought there was, the AMPA channel, that was the main excitatory one, and then the NMDA channel, that's another excitatory one, and then there was GABA A. That's the one that you can remember. That's the alcohol channel, and then GABA B. And there's a lot of other ones, but those are four biggies. So that was postsynaptic potentials, and those are basically how one neuron communicates with another neuron. It actually releases some neurotransmitter through the synaptic cleft that goes to the postsynaptic membrane and opens the channels, causes some current to flow. And so these included, like this guy was a sodium channel, this guy was a sodium and a calcium channel. The GABA A was a chloride channel, and GABA B was a potassium channel. So those, those are, like I said, those are the biggies. And then we talked about the special property of the NMDA channel. And so the NMDA channel has this property that it's both voltage gated So it's both voltage gated and neurotransmitter gated at the same time. And so that allows you to get something that's like Hebbian learning. And Hebbian learning was basically correlation. If you have a correlation between the input and output at a connection, that connection gets stronger. That's the Hebbian idea. But if they only get stronger, that's not good, because then you would just blow up, because everything would just get stronger. And so remember, we had spike timing dependent plasticity. And that, so how does the NMDA channel work? It works by, you've got glutamate, that indicates that the input line was active, and then you got depolarization, that indicates that the output cell was active. And if both of those occur, then the channel opens up. And so it can detect a correlation between the input and the output. That's NMDA. And that's the relation to Hebbian. So any questions about Hebbian? And oh, I forgot to start up the Zoom in case. Let me just start that up quickly. Is there any questions about that while we're waiting here? I'm not going to be able to do this quickly because I need to select the Mac. There we go.
Hey, Mark, you trying to sign in?
Yeah. Yeah, no, I figured out. There we go. Yeah, I got it. Okay. Yeah, so no questions about that so far. All good.
I have a question about the spike timing potential.
Yeah.
So about the spike, does the potential get strengthened when the spike just comes before the action potential, or?
Okay, so yeah, so the question was this spike timing dependent plasticity. How does that work? So if the input came just before the output became active, then that input helped cause the spike. And so that's good. So you get an upvote for that. If you just missed helping out, so like the output became active, and then you arrived just a little too late to help, then you get penalized. So you can sort of think of it as the causality detector. Did my input actually help cause the output to become active? If it did, then you get an upvote. If you just missed helping, then you get downvoted. And if it's off by about 1/10 of a second, just 1/10 of a second either way, nothing happens. So it's really like you just missed, then you get penalized. You just managed to help, you get voted up. If you're off by 1/10 of a second either way, then nothing happens. Yeah, so it's a very specific sort of timing going on there. It's kind of interesting how precise the timing has to be for that to work. So that was the spike timing dependent plasticity, and then that was essentially related to one more time, sign in. So that was related to our simple Hebbian learning models. And so our simple Hebbian learning models were looking at, let's just somehow simplify things down incredibly so that we essentially only have one input and one output. So now I'm waiting for my two-factor authentication. Okay. We're almost there. Sorry, guys. Okay. We are up. One last click. And yet one more last click. Okay, we're up. Okay, so where we were was we had, this is the actual physiology, but let's now simplify to this incredibly simple model to get some idea of like, what does that imply for a whole circuit? And it wasn't a complicated circuit. Basically, our circuit was we had one output, one input we're focusing on, and a couple other inputs. So it's just one output, a bunch of inputs. And the simplest way of stating a correlation is just that, or stating the Hebb rule, is just that the change in delta means change in weight equals input times output over many examples. So that's what a correlation is you just take a bunch from column A, bunch from column B, multiply them each pair, and then add it all up. And if you get a positive number, that's a positive correlation. If you get a negative number, that's a negative correlation. If you get 0, that's uncorrelated. But then we said, if you look at, what is the output? Well, the output is just, so the output just equals the sum over all the inputs of each input times each weight. So that's, so what a Hebb rule boils down to is the change in one weight. So the change in one weight is basically just proportional to that one weight times the sum of all the other inputs and weights. And so you can sort of think of that as basically it turns up one weight depending on the correlation of inputs. So the original Hebbian rule said change in weight is input times output. But since output is basically all the other inputs times all the other weights, a Hebb rule boils down to change in weight equals correlation between inputs. And so inputs that are correlated will turn up the weights of the correlated inputs. So that's where you can think of the Hebbian rule. A neuron looks out at its inputs and it says, who's correlated with who? Who's ever correlated, I'll turn up their weights. And so it just makes the neuron more sensitive to whatever the typical patterns that come into these inputs look like. So if you put a bunch of faces in there, you end up with a neuron that is selective for average features of faces. So that was our very simple Hebb rule. It's not a good Hebb rule. It doesn't look anything like a real NMDA channel or STDP type channel, but it just gives us some way of sort of thinking about what a Hebb rule implies. So input-output correlation implies pairwise correlation of inputs for changing weights. So that was our Hebb model. And if I ever ask questions on Hebb models, I always give you, just read the question. There's a bunch of information in the question. Just make sure you read the question. It will tell you how to do things. And I usually have some kind of simple problem on an attractor model. And so this model was just like a ridiculously simple model that didn't have any feedback connections. It was just inputs and then there's one output. There was no feedback at all. But what we said was, well, why don't we just like connect everybody up to everybody? And so then we have these little thermometer neurons, and basically they come back and connect to everybody. So if you have 3 neurons, there'll be nine possible connections, like connection here, 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 here. So there's 9 possible, including self connections. And so we said, in general, how do these things behave? And so how they behave is they have attractors. And what are attractors? You could sort of think of attractors as particular states And so what is a state? So a state is the activation, so unit 1 activation. And that going, and then this is unit 2 activation. So that thing is called a state space. It means like just the possible set of activations. So if you just have one unit, the state space is just a line. If you have 2 units, the state space is a plane, because there could be all different kinds of activations of those 2 units. If you have 3 units, then you end up with a cube. If you have 4, well, then you end up with a hypercube, but you get the idea. So it's just a way of representing all the different possible unit activation patterns that could occur. And the way attractor networks behave is they've got attractors. And so if you start off in some state that's not stable, it'll sort of head for the stable state. That's the meaning. That's why it's called an attractor. And once it gets to the stable state, it doesn't change anymore, even though it's still got feedback. So, inputs are still coming in, outputs are going out, but it doesn't change and so the activations are not zero, but the activations are not changing. That's the concept of a stable state. And so you could think of something, as an example, would be like categorizing something. You get all these different possible flowers, and then they all end up sort of activating the flower concept, the same flower concept. So that was our very simple model of sort of hooking everything up. And I usually have some kind of problem, but like I said, I'll give you if you have to update the network, I'll give you the rule of how to update it. So all I got to do is just read the question. It'll be in there. Yeah.
So if there were to be an attractor model on the final, just put 3 like you did, show examples for it, not the 2 like you did on the first example, that would be just
You could.
done so much better if it was 3.
It was 3. Yeah. Don't get too tied up to 1, 2, or 3. So basically the idea is the way you update the network is you just look at what the inputs are, and you just multiply the inputs times all the weights, and then you say, is it above or below 0? And then that tells you whether the output is like +1 or -1. And you can do the same thing with 2, or if you have 4. Yeah, don't get stuck on 3. 4 works the same way. So there's all different orders for updating. I'll always tell you, like update this guy, or just pick a random one and update it. I'll tell you I'll always tell you in the question how to update. Typically, I'll just say like, I might ask something like, is the network in a stable state? And then what you have to do is just update all the units and find, are any of them wanting to go to a different number than they are currently? That means it's not stable. If they all sort of like want to go to the same number that they're actually at, then you're stable. So that's really so it's a very simple model of sort of typically what happens if you just hook up everything up to everything. It sort of seems like it should be more complicated if you just hook up everything to everything, but in general, it will have this relatively simple behavior where it will, there'll be some configurations of the network, meaning some activation patterns in the network that if you update, they'll change, and then other activations that if you update, they won't change. Okay, so that was the attractor model. Yeah, we might have 2, might have 3. Yeah, they're basically the same algorithm. Just read the question like 10 times. It'll tell it's in there. It's in the question. Okay, so that's the attractor model. Then we talked a little bit about current flow in dendrites. So that was this, our model. We have like the outside of the cell is a wire like that, and then you model each little piece of the cell as a little capacitor and a little resistor. And then there's some goo inside the cell, and we model that by another resistor. And then here's another little chunk of the membrane, which we model by... The membrane's just one thing, but it behaves as if it's a capacitor, which can have charges stuck to it, either side of it. And then a resistor, which is like something that allows a little current flow through, but not much. And the main point here with this circuit was these resistance inside the network. So this is, the resistance longitudinal. That one was low. And this guy is the resistance membrane. That's high. So that means that most of the current will sort of flow down through the inside of the cell. Some of it will leak out, but most of it flows inside the cell. And then the wild card was the capacitors, and current can't flow through a capacitor, but some of the charges can pile up on one plate and then sort of be attracted to the opposite charge on the other plate. And so they serve as like a temporary little storage depot that could kind of complicate your picture. But eventually they get filled up at a particular voltage, and so then the current can flow, but they'll kind of gum up the works a little bit. And this was relevant later on when we talked about EEG and MEG, because this is what explains why you see EEG at all, because the current is flowing down, or either flowing down or flowing up in these dendrites that are all lined up. And so that's the fact that it flows a long distance inside the dendrite before it leaks out. That's the only reason you can see EEG on the surface. So it was actually kind of related to this big macroscopic thing, like recording EEG electrical signals from the scalp. Okay, so that was current flow in dendrites. You'd have to make a more complicated circuit if you wanted to include all the other channels in there, but we didn't talk about that. Okay, so that was the end of sort of low level. Then we started talking about the different modalities, and so we started off with viz. So visual, and we did a lot on visual because we're primates and we've got like a big visual system. So the first thing we did was the retina. And this introduced the idea that we have there's a couple things going on. So we have these formation of streams out of one source of information. And so you basically start off with the photoreceptors. And the photoreceptors are all off. So they're, in the sense that if you turn the light on, they get hyperpolarized. So if the light is off, they're slightly depolarized. If you turn the light on, they actually sort of get inhibited. But then what the retina does, it immediately divides this kind of one source of information into a bunch of different kinds of things. And so the first thing it does is it reverses the sign, and you get these on and off pathways. So the off pathway responds with inhibition to light. So it likes dark. So if you make it darker, it's happy. It's like the photoreceptor. But the on pathway reverses it. So if you make it darker, it's unhappy. It likes it when it gets light. So that's the first sort of split. But then each of these things split into this other typical split, which we see across many different systems. And this was, the so-called X and Y, or sustained and transient. So that's another common thing that happens across all different systems. You take a source of information and you split it into these more sustained signal, which just sort of tells you how much, and then transient, which tells you was there a change. And so that And then it gets, it gets further divided, like you end up with, red, and there's red and green pathways, and then remember there was a blue pathway, there was a separate, blue pathway, and that one got subdivided into, on and off. And so we end up with all these different, sub pathways, little streams, and that's something that happens in the other sensory systems too. And we even talked about, then there was yet another division, was lagged and non-lagged. Let me just put that up at the top, because that one might be relevant to let's go lagged, non-lagged. That might be relevant to the Reichardt detector, which was a way that you, remember that was a way that you could detector. That was a way to detect motion. And basically the idea is, it's a very simple idea. You just have one detector, and then it's got a delay. That's like the lagged. And then you got another detector that's offset. It's offset, in the X direction, say, and that guy is not delayed, and you add them together, and that's how you can basically detect a motion. So what happens is, a stimulus comes by, hits the first detector, that one gets a little delayed, and then the stimulus hits the second detector, then they both add up. So that was the Reichardt detector. And the problem with the Reichardt detector was the aperture problem. So if you set up a circuit like that, what's going to happen, it's going to have this problem in that if there's some object here, and the little detector, this little detector is set up right at that edge, it's going to have a problem in that it can't tell, what direction. All it can see is, it just sees something that looks like this. It sort of sees nothing, and then a little bit more, and then a little bit more, and then a little bit more. That's what it sees over time. This is over time. But if it sees something like that, it could be that, the edge was actually moving in that direction. But, the edge could have been moving in any direction, any vector that goes out to a parallel line to the edge. So it could have been moving in that direction. It would look the same through that little straw. And so that's the fundamental problem. We can't tell, is it moving that way? Maybe it was moving this way. Maybe it actually was moving that way. Maybe it was moving quite fast that way. Those all look the same. So that's the aperture problem. And in order to solve, so we can't solve this. If we just have a Reichardt detector, we can't solve this, because it's just ambiguous. It says it could be any of those directions. And so in order to try to solve it, we got to combine information across space. And so then we talked about, how MT could solve that problem. And how does it solve that problem? It looks at edges of different angles. And so if you look at edges that have different angles, then you can solve the problem. So that was and what's one way of thinking of the solution? It's, imagine you've got a detector that's trying to detect this pattern motion. And basically, if that detector gets input from all these Reichardt detectors that detect all these different local motions, if it gets a lot of votes, then it's probably that pattern direction. But it's going to be a sloppy system. So multiple neurons in MT are going to get a little activated. And the question is, which one gets the most votes? And that one's the one that's probably the actual direction. So, there'll be an overlap between these two. So here's another detector in MT. Say it's an MT, and say this guy likes that pattern motion. But the problem is there's going to be some patterns here that there's some local motions that are the same for both of those. But if you only got one edge, you're sunk. But if you've got, a bunch of edges, then you can figure out, is it this guy or is it that guy? So it requires looking at different parts of the object with different oriented edges. So then we talked about MSTD, and that was like a higher order aperture problem because MST can't see sort of rotation. MST can see like rotation and dilation, but MT can't distinguish those if you put the stimulus in the receptive field. And so MST has to do an MT-like thing again. It has to look at different parts of the stimulus in order to pick up the fact that there's actually sort of an expansion. MT can't see an expansion or a rotation. So that was basically, higher order. Okay, so then what did we do? So then we did maps. Yes, so there was maps. There's usually a question on maps. And so if we look at our brain, we've got the center of gaze there, and this is the vertical line, and there's the horizontal line, and then you have this funny thing in V2 where it sort of gets split open like that. And so that's basically on the brain. But then the question is, what is that, and this is a minus here and a plus there. So that's a little small. Let's make that a little bigger, sorry. Okay, that's a little better. So there's the center of gaze. There's the vertical meridian with circles. There's the horizontal meridian with the dashes. And then there's the horizontal meridian that's split in V2. And this was the plus was down and the minus was up. That's what the maps look like in the brain. But then the question, so remember that's a left hemisphere, so that's a LH. And what does that correspond to? That corresponds to the right visual field. So that's left hemisphere. But that corresponds to, the right visual field like this. And there's the plus and the minus. And so that's going to that. And so you can have a problem either way. You can say, here's what's in the visual field. What does it look like in the brain? But I could do a tricky problem where I say, here's something in the brain. What does it look like in the visual field? You can use the same technique to solve those problems. So if you have, say you've got like something in the brain that looks like a spot here and here, then what is that going to look like in the visual field? Well, it's in the lower field, and it's close to the vertical meridian. So here's the lower field, and here's close to the vertical meridian. So that's going to look like, a spot here. So you could go either way. Same technique. Like if you put the spot there, you could say, okay, so the spot is in the lower field near the vertical meridian. So I just go and look on this guy and figure out. You have to remember like V1 and V2. So here's V1 and V2. But if you can remember V1 and V2, then you could say, well, that I'm going to have one copy of that in V1, and then I'll have another copy of it in V2. Okay, so that was maps. Then we did simple and complex. So these were orientation detectors, just trying to detect the angle of an edge. And what did we do? So then we did a little bit on attention. So we said that attention wasn't in a particular place. It was just all over the place. It's basically a way of dealing with the fact that there's so much junk in the world. So when you look out, there's all this stuff all over the place. And we know that if there's multiple objects, they kind of compete with each other. And so they end up reducing the response of a cell. And we call that stimulus competition. So if there's like some little oriented object here, and if you put another object right next to it, the tendency is to actually reduce the response to that first object. And now the world is just filled with junk. And so the problem is all the stimuli are sort of competing with each other. And what attention does is it kind of suppresses all the other stimuli effectively so you can just sort of focus on one. It actually doesn't make the response to one bigger than it would be if there was nothing else in the world, but you can sort of think of it as what attention is doing is it's pretending that there was nothing else in the world. And then you get the same response you would if you just had only one object, because you get a big response if you just have one object, because there's nobody's competing with it. So it kind of rescues the competition and pretends like, oh, there's actually only one thing in the world. So it kind of basically suppresses the other stimuli. So, but it was all over the place. Every area has attention in it. It's kind of, and it's, there's different kind of attention in different areas. Some areas care more about what an object looked like. Other ones care more about, the movement of an object. Other ones care about the color. So you could have all different kinds of attention. So then we moved into the somatosensory system. So now we moved into the somatosensory system, and we started off with all the receptors. So there's all these different receptor types, a lot more different receptor types. So we had, gazillion receptors. So there's touch receptors, so just write them out. So we have touch, then there's a whole bunch of, X versus Y type touch, sustained versus transient type touch. And then there was pain, and this same thing in pain. There was like sustained pain versus like just change in amount of pain. And then there was temperature. We'll put that over here because it goes with pain. There was temperature. And then there was all the muscle receptors. But they're quite different. So we had ones that were doing length. Those are the muscle spindles. And then other ones that were doing force. And those two things are separate from each other. And the length ones, were broken up into sort of sustained and transient ones also. So we've got all these sort of channels of, streams of information coming in. But when they come into the system, they come in as maps. So there's still maps coming in. So you had all these receptor types, and then we talked about the maps. The maps are a little different because they're kind of all cut up into pieces, because your body, if you just look at your body surface, how are you going to flatten your body surface? I mean, it's like an incredibly complicated shape, so you got to sort of cut it into small pieces and patch it down onto the cortex. So they have more discontinuities in them than you have in the visual system. So an example was like, like your thumb is sitting right next to your chin. There's a long distance between your thumb and the chin, but they're actually right next to each other in the cortex. But they're still maps. And there was also a little talk about plasticity. So some of these maps can be sort of adjusted depending on if something bad has happened to some part of your body. So we talked about, and that's driven, we think that's driven by Hebbian learning, by actually sort of receiving information about what is next to what. So you could do it, bad experiments, originally based on human surgery, like take a little patch of skin from the thumb and stick it on the little finger, or vice versa. A little patch of skin from the little finger, put it on the thumb. You touch it, it originally feels like the little finger, but like a week or two later, it starts to feel like the thumb again. And so the whole maps have been sort of rearranged to take account of the new situation. Okay, and then we talked a little bit about, the connections of this. So remember, you got the ganglion cells, and they come in, and they go up to, the cuneate and gracile, and that goes up to, ventrobasal nucleus, that goes up to, area 3B. So that was some connections. Then we went to the auditory system. So the auditory system, we had the first thing is that you got this sort of 1D versus 2D business. So the auditory system is just, the cochlea is just a line of hair cells, basically. So what causes this? So what causes the sorting into frequencies? In a mammal, the main thing is the basilar membrane is varying in how rigid it is. And so the parts that are very rigid or stiff, those guys vibrate at a high frequency if a sound comes in. And the parts that are flabby, they'll vibrate at a lower frequency. And so it mechanically sorts sound into its different frequencies. And so, and what's actually driving it, you got these hair cells, and if you sort of tweak the hair cell back and forth, that's what causes the action potential to occur. But because there's only a line in the somatosensory system versus a two-dimensional sheet, then you have a tendency for a point to go to a line in the case of the visual system. This is visual somatosensory, but what happens in the auditory system, you only got a line, and so, but you've got a 3D thing to project into, and so that line will go to a whole plane in the auditory system. So that's odd. So put that a little closer. So that's the cochlea. So this is, retina or skin, and this guy is the cochlea, which is a line. So that's kind of a major difference between these two systems, but it uses this extra dimension for a lot of different things. And the first thing that we looked at was it does this kind of split into, X-like, which are sustained, and Y-like, which are more transient. And you see the same thing in the auditory system. And how does it do it? It basically, here's the hair cell, and here's the ganglion cell, and it either goes into, the potato neurons or more spread out neurons. And what happens is these synapses either just sort of hit these guys in different places, or they just go and hit just right on the cell body of the potato. And so this one is the time pathway, which is like Y-like, more interested in change. And this guy is the amplitude pathway, and that's more X-like. How much of it do you have? Not as sensitive to the change. So you can see that this division into X and Y is different in all. Here it's done in the retina. Here it's actually done in the receptor. The receptor is doing it in the somatosensory system. Here it's done by these different axon branch, different axon synapse pattern and different cells. So that was that sort of distinction between more transient response and a more sustained or amplitude-based response in the auditory system. Then we talked about binaural phase problem. And so the problem is, if we're trying to figure out like the time delay, one of the things that happens is we have this ambiguity if we just have one frequency. So if we just have one frequency, if that's coming into one ear and that's coming into the other ear, we don't know, like, should this wave peak be matched with this one, or should be matched with this one, matched with that? We don't know. And so that's a real problem in the system, because we notice when we look in the owl, the nucleus laminaris is binaural, meaning it gets input from both ears. What does that do? It just matches everybody. So, spikes come in this way, spikes come in this way, and here's the nucleus. It just says, I'll just match any old spike that's coming in, with the spike coming in the other way. So this guy's coming in that way. These guys are going in this way. So here's the cells. It just detects coincidence in spikes. And if you do that, that's equivalent to matching all these peaks. And so it's ambiguous. So how would it be ambiguous? Well, this guy might be activated, and then also this guy might be activated. And so that's equivalent to, like, you match this guy with this one, and you also matched it with that one. And so how do you solve that problem? You have to do something like the aperture problem. You have to look across frequencies. And so if you look across frequencies, then you can find out what's the real delay. It will be the one that sort of lines up across frequencies, and that was a picture like this. So if you just have one frequency in, say, the inferior colliculus, central nucleus, lateral part, if you just have one frequency, there'll be an ambiguity if frequency is going that way. But if you have another frequency, say a higher frequency, it will also be ambiguous. Might look something like that. And if you have a lower frequency, it will also be ambiguous. And so you'll end up with, something that looks like, one column, that's the real delay, but then there's another one that's kind of bent off because it's different delays at different frequencies. And so then what you can say is, that's the one, because all that lines up across frequency. And that's also a mechanism how you could attend to something. If you just turn up that column, then you'll blot out things that are coming from different parts of the world. So that was the auditory system. And then we talked about bats. So then there was bats. And we talked about the Doppler shift. That was, due to the bat is, all the stuff is moving that it's trying to catch, and the echoes will come back with a shift in frequency. So we talked about the Doppler shift. And the bat's voice is going all over. The bugs are flying at all different speeds. But if it wants to figure out how fast the bug is going, it has to sort of calculate the Doppler shift. But it has to calculate this Doppler shift between, and ignore whatever the absolute frequency is. And so that was good for essentially, bat, calculate Doppler. But it was equivalent to this problem of, vowel across vocal tract size. Because we're trying to just figure out somebody saying an E, but I don't care how big their mouth is. I just want to know, is it an E or an ah? So that's kind of the same problem, because somebody with a smaller mouth will have higher frequencies for their E, but they'll be spaced in a characteristic way. And so you're just trying to detect that spacing. Is it far apart spacing like an E, or is it closer spacing like an ah? But, you know, a small person would have an E up here, whereas a big person would have an E down here. So that's the same kind of problem. And then there's also like delays, you know, because the bat has to figure out how far away something is. Doppler doesn't tell you how far away it is. It just says, what's the speed difference? So it has to do delays. So then we went to the superior colliculus. Okay, so let's go to motor. So the superior colliculus is kind of like half of the motor system. So superior collic. So for that, we said we had a retinal map. It's kind of a layer cake with 3 layers. So we had a retinal map at the top. This guy is retinal. And then motor map at the bottom. And then in between, we had something that we said was like a saliency map. Now, so what's the point here? So the point here is we're just trying to look at stuff. There's a bunch of stuff out there that comes in on the retinal map. So here's, you know, 1, 2, 3 comes in on the retinal map. And what do we want to do? We want to just pick one and look at it. And so when you pick one, what happens is we send some activity down to the motor map, and that makes a saccade that will get our center of gaze, the center of the retina, on that thing. So that's relatively simple if you just have multiple objects that are on all the time, like 3 little LEDs that are on, no problem. But the world isn't like that. The world is just like stuff's changing and how relevant it is to look at all the time on a half-second by half-second basis. You know, something moves, it's very interesting, but then it stops moving. But it still is interesting to look at that even though it stopped moving. And so that's what the saliency map is supposed to be. It's something that sort of buffers or sort of manages how interesting different parts of the environment are over time. And it doesn't just rely on the fact that that object is interesting at this point in time. It says it was interesting like, you know, a couple seconds ago, and so I'm going to keep track of that object. That's what the saliency business is. And so then, you know, we talked about the problem of managing the saliency map. So the retinal map is no problem because there's just stuff on, and if stuff stays on, then you could you know, at a later point in time, you know, look at where. So let's think what happens as soon as we move. As soon as we go down into the deep layers and move the retina, we're moving the retina. What's going to happen? It's going to shuffle all the targets around because now they're in a different place because you moved the retina. If the target stayed on, no problem. Then they'll move to some other part of the retinal map, and then we can pick one of them and trigger the deep colliculus and get the proper eye movement. But the problem is, what if they go off? And so that was when we talked about this double step. And double step is just kind of like a very simplified way of trying to state the fact that everything doesn't remain equally salient all the time. It's sort of like somebody does something that's interesting, and then you go like, I'm interested in that spot, but then they stop doing whatever was really interesting, but you still want to look at it because they might do something interesting again a little bit later. And so the double step saccade is you just put up 2 targets and turn them off, and then the animal has to look at both in order. And the problem there is if they're off, we can't rely on the retinal map as to where they would have moved to. And so how do we describe this? So we describe this as hallucinate, where targets would have gone to in superficial layer if they stayed on. If they had stayed on. So that's basically how you solve this problem. You just simulate where they would have gone to if they'd stayed on, even though they're no longer on. And what does this? It has to come from some eye position signal. Eye position. So eye position signal has to go up into the saliency map and move stuff around. So it's constantly doing this, day in, day out, every 5 seconds, every 100 milliseconds. It's just updating, like, where is the interesting things to look at. Every time your eyes move, the eye position gets fed back into the system, efferent's copy, and it moves all the targets around, whether or not they stayed on. That's the hallucination part. So then we gave some examples. So the one example of it was this double step, but we gave another example of what happens to auditory targets. Auditory targets. So say you want to look at an auditory thing. Some sound happens. It's a very natural thing to do. Some sound happens someplace, and I'm attracted to it. I need to sort of look at that location where the sound came from. Now, the problem is that your eyes can move relative to your ears. So your eyes can move all around, but your ears are kind of stuck to your head. They can't really move. I mean, your head can move too, but your eyes can definitely move differently than your ears. And what's the problem? Well, your ears haven't moved, but your eyes have. And so if your eyes have moved, it's going to move all the targets on your retinal map because your eyes are in a different place. But your ears are still stuck to your head, and so they're still reporting where a sound is. And so what happens? The map location of the sound gets moved. So with auditory targets, eye position, used to move the auditory response to, line it up with retinal map. So if you're looking straight ahead and there's a sound that comes sort of near the center of gaze, you need a small eye movement. But if I'm looking off to the left, that same sound now has to make a larger eye movement, which means we've got to move it to the place in the retinal map that codes a larger movement. So that shows you that what is the colliculus doing? It's just trying to look at stuff. And so it doesn't really, it has to somehow fix the problem that the retina can move around, but the ears are stuck to the head. And so how does it fix it? Well, since this whole motor map is basically a retinotopic map, or a retinal map, it has to move the auditory response around to sort of take into consideration where the eyes currently are. It uses the eye position to do that. So this shows that the system is trying to maintain a coherent representation of the world for the purpose of looking at stuff. But we had another sort of inverse version of that when we looked at VIP, that's ventral interparietal. This was kind of the opposite kind of situation. So in this situation, what is VIP for? One idea is that it's kind of to avoid getting hit in the head. So if something's flying toward you like this, you want to move out of the way. But the problem is your eyes can move relative to your head. So maybe I'm looking off to the left and something is flying toward me, or maybe I'm looking off to the right and something is flying toward me. In that case, the visual data is going to be all over the place. It might be in the opposite hemifield. And so what happens in VIP is kind of the opposite of what happens in the superior colliculus. You know, the same information, the eye position. So here it's moving the auditory response. Eye position used to move visual. It actually moves the visual response to, line it up with the face touch map. So the face touch map doesn't move. So, the face touch map stays in the same place. And what happens, because what's the goal of VIP? It's just to sort of don't get hit in the head or bite something. Bite something, but don't pay attention to where or somehow factor out where your eyes are looking. So if something's coming toward your mouth, you got the fork coming up toward your mouth, you want to bite it. Maybe you're looking up to the left, maybe you're looking up to the right. You don't care. You want to know where the thing is. You want to know where the visual thing is relative to your mouth. And to do that, we've got to somehow factor out where you happen to be looking. It's kind of the opposite of what happens in the superior colliculus. Okay, so there's a lot of other things like this going on having to do with arm movements. We didn't talk about any of that stuff, but this was two well worked out examples of what's the goal. The goal is to avoid stuff hitting your head in this case, or reference things relative to your face. The goal in the superior colliculus is I just want to get my center of gaze on the object. Okay, so then we went to the whole rest of the motor system. So, and for that, what did we talk about? We talked about pattern generators, and that was this idea if we, like I always give the cat example, so like a cat sort of batting at something, batting at a toy. And so here's the cortex, and the cortex says to the pattern generators, just run your bat at a toy program. And then these guys control the actual motor neurons. Now, the problem is, if the cortex doesn't have any detailed access to it just says, like, just do your batting stuff. That's going to be like 20 muscles. So, the pattern generators take care of all the, all the coordination between all those muscles in order to get an effective bat. But what happens what happens if you want to, like, play the piano? And there's no pattern generator for playing the piano. And so what's happened in multiple times in songbirds or raccoons or monkeys for controlling their fingers is the cortex sort of skips this and projects directly to the motor neurons. And what does that do? That will, primates, raccoons, birds, songbirds, what that will do is that will allow the cortex to be at a lower level. So the cortex can now sort of coordinate the individual muscles, all these individual muscles, directly, and do more complicated things. So that was sort of bypassing the pattern generators and moving basically moving the lower level pattern generators up into the cortex. Now, you leave the pattern generators there because, a monkey still has to do batting kind of motions, and so that circuitry is still there, but you got another pathway. So that was the pattern generators, and then there was the cerebellum. So there's lots of stuff in the cerebellum, but the part we want to remember, which is good to remember for the test, is the cerebellum and the striatum they're like these side loops that help gate particular actions. So they're inhibitory. So the output of the cerebellum and striatum are inhibitory. And what they're doing is when they ease up on the inhibition, they allow a motion to come out. So they act like a gate. And the main thing that I want you to remember is just where is the cerebellum? So the cerebellum, so the cerebellum gets some input from the spinal cord, but it gets input from the cortex. But how does it get to the cortex? Well, it goes to the pontine nuclei. So say MST, for instance, projects to the pontine nuclei, and then that will go into the cerebellum. So let me see how many steps I put last year. Not too many steps. Yeah, so I'll put them both in there. So this will go to the granule cells. So now we're in the cerebellum. And then the granule cells project up to the Purkinje cells. And then the Purkinje cells project to the deep cerebellar nuclei. And then the deep cerebellar nuclei project, up to, the red nucleus and the thalamus. And then, the red nucleus can go back out, and the thalamus can go up to motor cortex, and then back out. So it's important to just remember, what's the main picture here? And of course, the cerebellum is going to get a lot of sensory input too from the spinal cord. But the basic plan is you have this little side loop where you can go into the cerebellum, come out of the cerebellum, and then get back into the motor cortex. Now, there's other stuff, moving the body around, but it's important to just remember the order. Cortex, pontine nuclei, you go into the cerebellum, you go to the deep cerebellar nuclei. Those can go and then get to the motor system either directly in the red nucleus, immediately back out, or go up to the thalamus and then go up to motor cortex and back out. So that's how the cerebellum can affect a movement. And then we also talked about the striatum. And the striatum, you have a similar set of things to remember with the striatum. So the cortex, it's another gating system. And so the cortex will go down to the caudate and putamen. Those are kind of similar kind of things. And then that goes to the globus pallidus. And then the globus pallidus, which is in the forebrain, remember, because the globus pallidus and caudate putamen are part of the forebrain, will go down to the motor thalamus. Then the motor thalamus will go back up to motor cortex, and then that's how you get out. Motor cortex out. So it's important to just remember these kind of basic circuits on how these kind of side loops of the cerebellum and striatum can influence motor behavior, because many other things are influencing motor behavior, not just the cerebellum and the striatum. So the motor system has all its own circuits, but it's got these kind of loops where you go through the cerebellum, come through the deep cerebellum, and then up to the motor system, or from the striatum, you go through the caudate, putamen, globus pallidus, and then get into the motor system through the thalamus. Okay, so those are just basic sort of connections that every college-educated person should know. Okay, so that was the striatum. And in the striatum in particular, we talked about this kind of temporal hierarchy. So, so there's some parts of the striatum that are hooked up to, the limbic system, and they might They might involve like goals that are taking place over, over an hour. I'm hungry now. It's a long-term thing, like, go get some food. But then you have other very local goals, like just pick up this thing here. That only takes like a half a second. And so the striatum seems to be involved in kind of segmenting behavior, but different parts of it are involved in segmenting behavior at different temporal scales. So sometimes you've just got like an urge to do something for an hour, but then in order to do that, you've got a whole set of little programs that have to be played out, and the striatum is involved in both of these kinds of things. So that was the striatum. Then we talked about the limbic system, which you can sort of think of as another motor structure, because it has access to hormones, and that can really cause your body to do a whole bunch of different things. And but it also has access through the pedunculopontine nucleus to actually sort of moving you around. It's like another way to get into the motor system. And in general, the idea there was basically we want to somehow, mediate between. So here's the limbic system. So it can mediate between, like the cortex, which knows how to do stuff, and it can sort of directly activate the body through hormones, or it can sort of more indirectly activate the body through, the pedunculopontine nucleus, and that can go up to the cortex and back out. But you can think of it as basically monitoring internal state, like hunger. But not just hunger. So you have things like, hunger and thirst. But you also have head direction. And weirdly, head direction, it's an internal state. It's what direction my head is in. And place, where am I at this point? So, and at least half of the limbic system is involved in this kind of, where are you in the environment? What direction is your head pointing relative to different cues? And so we went over, all the head directions, place cells, and that whole system, which is in the limbic system, mixed in with very close to like hunger and thirst and sex drive and all that stuff. But it's basically a system for sort of, one of the functions of the limbic system is a system for sort of representing all this internal state, like, how hungry am I, where am I, what direction is my head pointing at this exact moment in time. And so we talked about all the cool experiments with place cells and head direction cells, and how they use, distal versus local cues. So distal cues were like, clock on the wall, local is like a little P on the paper. And, distal cues are preferred because those are more likely to sort of be good cues about figuring out where you are. Okay, so then, just about 5 more minutes, we, last but not least, talked about EEG and MEG. And we said that there's 3 things you need to make a signal. One, you've got to have, you have to have anisotropic dendrites, meaning like they're sort of extended dendrites in one direction. And then they have to be aligned with each other, like pyramidal cells are. And they also have to be layer-specific activation, because otherwise, if they're all activated different places on their apical dendrite, you won't get a signal. But if you have all of those, then you get a visible signal at a distance. So that's basically what causes EEG. So it's basically going back to the dendrites here. There's conductive goo inside, and it's hard for the current to leak out. And because of that, coupled with 1, 2, and 3, you end up with a signal. So that was EEG and MEG. And that's more related to actual stuff going on. It's relatively hard to localize compared to MRI. And so a lot of people have gone to MRI. It's slower because what are you measuring here? For functional MRI, we'll put an F in front of there, you're measuring sort of blood oxygenation. You're measuring blood oxygenation, in response to activity. So the brain becomes to neural activity. Some neurons become active and they immediately demand more oxygen. So that's actually what you're typically measuring. The blood oxygenation change. Kind of slow. It takes a couple seconds for that to happen. But it has really good spatial properties. And then we talked about the Bloch equation. So Bloch. And what was the Bloch equation? It was, how does the magnetization change? How does the magnetization vector change? And there were 3 parts to it. The first part was we take the current magnetization vector and cross product it with the magnetization caused by the magnet. Those two are different. And that causes this precession business, so that first one caused the precession. And then we had another one that looked at the XY magnetization divided by T2. And that one sort of was the one that got rid of the X and Y magnetization quickly over like a 20th of a second. And then we had another one that looked at the Z magnetization. But remember, it was the Z magnetization, it was MZ minus MZ0. Oh man, I'm off the edge. It was the difference between MZ minus MZ0. MZ0 was like what the magnetization gets up to after you've been in the magnet for a while. That was a T1. And so what are those things? This thing does the precession like a top. This one does, transverse decay. Quick, that's, fast. Yeah, and this one does longitudinal, Z, regrowth. So if I ask a question about the Bloch equation, sometimes I ask questions like, which things are constant here? Like the B is not constant. That's movable because that's what the magnet is doing. The M is not constant because the M is actually the local magnetization. That's changing, is decaying. T2, that's a constant. That's just a number. So I might ask some question like that. So there's the Bloch equation. And then last but not least, I don't think I've ever asked a question on this. There was the Fourier transform. But the main point there was as the gradients wrap up spins into stripes. So if you turn a gradient on, what it does is it sort of makes the ones that are at the high end of the gradient go faster, and they get several cycles ahead. And those stripes are basically, one term in Fourier transform. So that's basically how you make an image. And so you turn on these gradients, they cause these stripes and spin, and then collect a whole bunch of different stripe patterns, and that's also known as a Fourier transform. And then you just un-Fourier transform it. So this is how you get an image. So the gradients are really what you need. The important point here was the gradients are what's doing that. Okay, I think we've covered the board. So any final questions here for the whole course? All good? Yeah.
Is the only thing that's constant in the bucket system T2, or is there anything else that?
No, T1 is a constant. Yeah, T1 is a constant. MZ0 is a constant because that's not a variable. That's just MZ0 is just what the magnetization gets up to after you've been in the magnet for a long time. So that one doesn't change. That just depends on number of protons, just the number of protons you have. And so that's a fixed thing. Well, I mean, it's not constant if you put a different head in there, but for a given head in a particular position, it's a constant. But the amount of Z magnetization, that varies. The XY magnetization, that varies. That little gamma there is a constant. This thing is not a constant because, over time, the change. So like this, this is the change in the magnetization. The change in the magnetization is going to change over time. So that's not a constant. And then the B, remember the B here, that was the main magnetic field, and it was the RF coil flipping the magnetic field around, and it was the gradients changing the Z component. All those were mixed into the B. Okay. The finals, yeah, okay, yeah. So let's. I'll send out another. It's at 8 a.m. Yes, I can't control that. That's the final schedule. So Monday at 8 a.m. And so it's about half old stuff, half new stuff. And there's 12 questions instead of 8 questions, but you got more time. So there's a lot less time pressure. And it will be here at 8 a.m. No, 2 hours at least. Yeah, 2 hours at least. Yeah. No, it's less time stress. Okay. Well, see you guys on Monday. If there's any questions, I will be checking my email over the weekend. I can respond then.